much, again, the Noldus team and Robert and colleagues. Well, the problem we have in the field is that, well, of course, we will complain about, we don't have good models, we don't have good tools. This is very true. So, one of the strategies in biological psychiatry research in order to improve the phenotyping is to actually increase the spectrum of model species that we use and apply that new knowledge to understanding brain disorders. That's one approach. So we're good on this domain using zebrafish as an alternative new model species to complement the rodent research. And the second is to approach is to maximize the phenotype based data so that we can get more data per experiment, more data per animal, more data per trial time. And this is very important. This is also a strategy to go. Now, I'm going to talk about how to do this using zebrafish, adult zebrafish, but many things can be applied to larval zebrafish as well. We simply don't know, we never tried, but maybe that can inspire interesting collaboration. All right, now I want to talk about one specific success story, and this is this very unique drug called ibogaine, which is, in my view, is the king of hallucinogenic drugs. Well, the reason for that statement is because of the unique profile of this drug. This drug works at serotonergic pathway, glutamatergic pathway, opioidergic pathway, meaning that it works as LSD, ecstasy, mescaline, phencyclidine, psilocybin, ketamine, salvinorin A, all combined. So you would expect some sort of very unique profile from the drug like this. And indeed, this is what we have. Now, just a little introduction. This is a kind of evolution of the approaches that we have in our lab, and it kind of reflects the overall level of approaches that the field uses. So this is hard-working students recording zebrafish, being dedicated. Well, we know, of course, that they want to get to medical schools. So that explains the passion. Now, zebrafish doesn't have to go to medical school, but it's curious. So then you can have a little tank and a pretty cheap web camera connected to a very cheap laptop, and then you have the studio. We call it zebrawood in our lab, but you can call it however you want. And it's very kind of space efficient, economical, and you get something like this. And of course, you know that zebrafish like to spend time at the bottom, and when they're getting curious, they go to the top. So these are the rules of zebrafish behavior. Now, then we decided, okay, well, unlike mice, zebrafish, aquatic species lives in the water. They have three-dimensional behavior, so we should do something about it. We should capitalize on their ability to swim in three-dimensional space. So we started using three-dimensional analysis. The endpoints you can get by manual observation are listed here, all based on common sense: time to top, number of entries, time and top, erratic movements, freezing, time frozen. But you can also use zebrawood to extract the additional derivative endpoints, such as duration of swimming, distance traveled, angular velocity, average velocity, up to several dozens of endpoints. So this is good. So we can get more data, more endpoints per animal. Well, this is just to illustrate how that works. You give fish fluoxetine. Fluoxetine is an antidepressant. Chronic fluoxetine is an anxiolytic antidepressant in mice. In humans, and now you see the same pattern in zebrafish. So you see that they don't go to the bottom much, they go straight to the top, spend a lot of time there, distance traveled is not affected, so it's not a psychostimulant effect, average velocity not affected. Behavior, erratic behavior goes down, cortisol goes down as well. So behavior follows physiology, you have a good story to tell. Well, yes, but the problem is, if you do use many endpoints, that's what you get. The multiple bars that change in all possible directions, and you cannot understand what they actually mean. Now, what we thought is important is to actually try to visualize what zebrafish do, what kind of behavior they show. And the only way to do it, we believe, is by analyzing and documenting, cataloging their three-dimensional behavior, their behavior in X, Y, Z coordinates, and also X, Y time. So we use that setup, 2 cameras. This is a novel tank. This is dedicated zebrafish swimming quite happily. And this is where the Noldus team came to our help, offering a new software. I don't know if it's available for the clients or not, but colleagues say that it is. So it does this analysis for you to generate the three-dimensional traces where the velocity, the angular velocity, all these endpoints can be calculated based on three-dimensional fish behavior. So this is a typical control. This is an example of how calibration is made, just to make it very accurate. And this is the control zebrafish, wild-type zebrafish swimming in the novel tank. You see a lot of time they do spend at the bottom, and they do occasionally go to the top because they are curious. Well, this is a different story. This is not even ibogaine. This is nicotine. So nicotine, as you could see, very potently affects zebrafish behavior. In 3D, that's what you get, and then you can use color to denote velocity or angular velocity, and that enables a dissection of different fragments or parts of zebrafish trajectory if you want. And why is that important? It is important because you can teach the software to detect those thresholds, so that no longer the students or the postdocs have to do this. The machine can detect the endpoints, the phenotypes of zebrafish, including very complex and very difficult to manually detect, such as erratic movements. All right, this is another example of LSD. LSD, as you could see, compare it to, let's say, nicotine, right? A completely different picture, where velocity changes more radically, and there is no stereotypic kind of swimming along the periphery. It's something completely different. In many ways, the predominant effect is uncorrelated, but you see the difference. You see that difference only because it is visualized on a 3D level. And then just for the record, these traces are not the best that we get. These are the median, meaning that they are representative of the phenotype to the best of our abilities. And we do ranking by 3 individual observers independently, and then based on the consensus. All right, now this is back to ibogaine. So this is what ibogaine does to zebrafish. We're talking about the acute exposure to 20 or to 10 or 20 milligram per liter for 20 minutes. So we put fish in the beaker, exposed to ibogaine, and then test them in the novel tank. So the controls, as you could see, this is XY time is here, and this is XYZ. You see a typical pattern of fish going to the top, to the bottom, staying there, and then eventually going to the top to explore. Makes perfect sense. Well, ibogaine does completely the opposite. They go to the top, quickly explore the top, and then they go to the bottom right after that. So it's a reversal of the normal zebrafish behavioral response, which we found very intriguing, very unique. We have never seen anything quite like that. Well, maybe that's what you would expect from the king of the hallucinogenic drugs, something that you have never seen before. Right, just to give you another example of another fish. No, this is interesting. So this is another way, very intuitive, to visualize the data. So each cohort, we usually use 12, 15 zebrafish. So this is what you get when you superimpose on one diagram the traces x, y time for all 12 fish. This is control. And the color reflects, I believe, velocity. But that is not that important. Just look at the shape. No pattern whatsoever. And then here you have traces of ibogaine that all 12 fish look pretty much the same, like a waterfall, which is very illustrative, and it actually raises an interesting discussion. We can come back to that later, whether this behavior is more synchronized by ibogaine as compared to less synchronized control fish swimming. So this is something to consider, but this is just to give you an idea of how robust, how strong phenotype is. Now, another interesting behavior is home-base formation. So the zebrafish, like rats and mice, they do establish safe locations, the home bases. But what is interesting, they all do, but what is interesting, look at the control. They do establish a home base. I would say this fish would do it here. So this is x, y, time. Time goes that way. And this is kind of like the top view summary, and you can see that, yes, this is the home base. But look at the ibogaine-treated fish. They establish the home base and stay there. They do explore other parts of the tank, but it looks like it doesn't matter for them. It changes their spatial strategy, their spatial awareness. And, by the way, we're talking about translation research. This is what humans talk about describing their personal experiences when they have ibogaine experience clinically. So it is interesting and striking, and the analysis here at the bottom actually show that, yes, the home base formation is present, but it's much more faster and much more pronounced in the ibogaine-treated fish. All right, down the alley of my colleagues, other speakers, the social behavior, the shoaling, is affected by ibogaine as well. You could see the control fish, relatively tight schools, and the ibogaine-treated fish, kind of like not so much. Now, what you may not see here is that actually the color of the fish changes as well. And this is where ibogaine, we think, is quite unique, although other drugs have done that too. So, if the lights are switched off, you will see that the controls are much more pale as compared to the ibogaine-treated fish that became very dark. Now, we did that subjectively by using a simple scale, 1 pale, 2 dark, and the reviewers didn't like it, so we said, okay, we'll do it right. We took photographs, converted that into pixels, used the software to analyze the pixelations, and no kidding, the coloration of the ibogaine-treated fish was darker. Reviewers were happy, we published a paper, and it worked. Now, we're talking about not even behavior, we're talking about skin pharmacology. I wish mice can do, I wish mice can do that, they cannot change the color. But just think about this. You screen the compounds, and if you see a tank where fish is dark, it means something. It's a phenotype of interest that we can extract and consider. So sometimes testing different drugs using zebrafish can give you interesting surprises. But as good, or at least striding scientists, we try to quantify and measure it and try to make any sense. All right, so some glutamatergic drugs also do this as well, which makes me think that perhaps this could be a good indicator of glutamatergic antagonism that ibogaine has, and ketamine has, and tencyclidine has, and they all make fish darker, but ibogaine, I would say, is much more potent. Well, I would skip the videos. I show the graphs, and I would like to thank the audience, and my take-home message is watch this animal, model species. Zebrafish research rocks. They will continue to rock, and we are to see a major expansion of the field. So the sooner you join the battle, the better it will be for your RO1s. Thank you very much.